So glad you all came, and I hope you'll learn some new things from this presentation. So I did research over the summer at Sam Houston State University, and it's regarding, as you can see, chromosomal replication related gene organization in Rhodobacter sporoides. So I'll start off by saying like how you can get internships. So I think there are two ways. You can either contact faculty at a nearby university. You can go to their university page, look at what research they're doing, and then send a nice email to them asking for research opportunity with them. Or you can also apply through programs that will grant you research opportunities such as Welch. So those are, I think, two good ways to get into the world of research. So, and my mentors, I would also like to thank them. They're Dr. Chowdhury and Dr. Cho. So just for an introduction, I'll just explain some basic things that will help you understand our research. So first of all, let me explain to you the field of bioinformatics. So bioinformatics is kind of the bridge between computer science and biology. So we use computer science programs to analyze things such as genomes, which I've done in this presentation. So yeah, it's a new and upcoming field in the field of biology. It's also a component of biomedical engineering. So for those who are interested, it's a very cool subfield. So why did we choose Rhodobacter sporoides? So we chose Rhodobacter sporoides because it has versatile growth. That means it grows in anaerobic, aerobic, and photosynthetic conditions. So we know that it's very diverse and we won't have any problems with growing it. Also, quite uniquely, it has a multi-part-type genome. So usually when you think of bacteria, we think of one circular chromosome. However, Rhodobacter sporoides has 2 circular chromosomes and 5 plasmids. So the first chromosome is about 3 megabase pairs, and the second one's 0.9 megabase pairs. So that's a very interesting feature of Rhodobacter sporoides. So I'll just go in brief over the method of chromosomal replication. So in the top picture, you've probably seen that in your biology textbooks, because that's where I got it from. Yes, so you can see since DNA polymerase can only construct a fragment of DNA from the 5' end to the 3' end, like the new strand, that creates a scenario where you have a leading strand, which is assembled continuously, and a lagging strand, which is constructed semi-discontinuously. So it's constructed in little portions, but it's still ultimately going to either the right side or the left side. So in the second picture, it's just showing another picture of DNA replication with DNA polymerase, and then you also have primates and helicase as well. So I mean, so we have the right replicor, which goes the right way, and the left replicor, which goes the left way, and then you have the origin in the middle, where there's the origin bubble, and then you have the terminus about opposite to that. So our research is concerning the asymmetrical distribution of genes throughout the chromosome. So when I was learning biology, I always thought that the genes were evenly distributed. There wasn't any like bias for what strand the genes would be on. But through this research, I learned that that's not true. There are certain parts in the chromosome that have more genes than other chromosomes. And so in talking about that, the model that explains this is called the collision model. So collision model, that's a diagram right there showing the 2 types of collisions that can occur between DNA polymerase and RNA polymerase. So in the first one, we have a head-on collision. So that's when you have an RNA polymerase and a DNA polymerase. They're transcribing or replicating. So they have a head-on collision, but that leads to an aborted transcript. And that aborted transcript is not functional. And it actually can be harmful to the bacteria. However, if you have a co-oriented collision, that means that they're going the same way, but they just collide due to different speeds. And that produces a complete transcript. So ultimately, you want a complete transcript, so you have a functional RNA sequence, so you can have a functional protein. And that's why co-oriented collisions are preferred. And so that's why genes tend to be on the leading strand of the chromosome rather than the lagging strand. So you have more genes on the leading strand. And one of the factors in the asymmetry throughout the chromosome. Another factor is the concept with essential genes. So in this research project, we classify essential genes as genes involved in transcription, translation, and replication. So these essential genes, while they tend to be on the leading strand, they also tend to be closer to the origin. That way they have a higher chance of being successfully transcribed, and so they have a higher success rate at expression. So our two hypotheses for this research project were, first one was the ratio of the number of genes on the forward to the complement strand would be close to 1 at the replication origin. And that diagram shows you why. So the bold red line is the leading strand, and the dotted is the lagging strand. So at the origin, it switches from leading to lagging, or vice versa. So that's why you have an even distribution of genes at the origin, and so you have a forward to complement strand ratio of approximately 1. And it's also approximately 1 at the terminus. However, on the right replicor, or the right half of the chromosome, you have a forward to complement strand ratio of more than 1, because if you see it as a fraction, forward to complement, the forward strand is the leading strand, which has more genes than the complement, and that's why it's more than 1. And on the left replicor, it's less than 1 for the exact opposite reason. Our second hypothesis was that the highest percentage of essential genes would be in the flanking regions of the chromosome replication origin. So near the origin, as I explained, to increase the chances of successful transcription. So I'll talk about my first, we did 3 methods in total. The first 2 are for hypothesis 1, and the third method is for hypothesis 2. So for method 1, before we put method 1 to use, we did some baseline calculations. So first we used OriFinder, which is a readily available software, where we input the genome of the bacteria, which is completely sequenced and marked, and you can find that on NCBI. So we entered that into OriFinder to get 3 putative replication origins. Putative meaning we don't know which one, but it could be one of them. So 3 for chromosome 1, and then 5 for chromosome 2. So once we got the putative origin, replication of origin, we can test each of them and see which one's the functional or the real one. And also I did some baseline Excel calculations as well, which took a very long time because I had to take PTT tables. So PTT tables are protein table something. It's the complete sequence of all the genes in the chromosome with their product and what protein they ultimately produce. So I had to put that into Excel and use a lot of formulas, a lot of conditional formatting and stuff to get some basic numbers, to get what percentage of genes are on the leading strand versus the lagging, and such calculations like that. And I got approximately like 52% for leading to lagging. So Rhodobacter sporoides surprisingly has a very low bias for the leading strand. And I must say that took a very long time and many, many hours staring at the computer screen. My eyes started squinting after a while. Yeah, but it was all worth it. So after that, I'll proceed to talk about our first method. So the first method involves the flanking regions analysis. As you saw in the previous slide, you want a ratio close to 1 at the origin. So what we did is we took 5 bacteria with known origins of replication and did the same analysis on them. And as you can see on the rightmost column, you can see that the forward to complement ratio is 1, 1.1, 1, 1.3. So it's very close. So that way, we know what to compare. And then we tested the 3 putative replication origins for chromosome 1 and the 5 putative replication origins for chromosome 5 using the same test. And for the third putative replication origin, we got a number of 1.188, which is quite close to 1, and the closest to 1 of all the putative replication origin. And then for the fourth putative replication origin for chromosome 2, we got 0.916, which is also quite close. So this method is one way to verify that that site, as you can see the coordinates, those are the base pairs. So that site is the functional replication of origin. Our second result also was to prove where the replication of origin is using forward to complement strand ratios. So in this, we did a sliding window analysis. So it's a bit difficult to explain, but if you imagine that chromosome right there, and you start at the origin, a sliding window analysis is where you're having cumulative increasing segments. So we did it by 20 kilobases. So the first bin we tested was 20 kilobases. The second was 40, the third was 60, and etc. So it's cumulatively increasing. And so using that, we generated this graph. And as you can see, so at the zero mark is the replication of origin we think it is. And as you can see, so the blue is where it's above, the forward to complement ratio is more than 1. Then it goes into green region, which is less than 1. And then it shoots up quite a lot to be above 1. And that's where the origin is, because if you see in that diagram, see, it's more than 1, and then it becomes less than 1, and then it becomes, and then it immediately shoots up. So that's where the origin of replication is, theoretically. And we got the same result for chromosome 2. Now you may be wondering, why are there 2 places where it switches over for chromosome 2? So we believe that chromosome 2 is a rather recent chromosome in the evolutionary history of Rhodobacter sporoides. So it originally originated as a plasmid, and it recently became identified as a chromosome. And that's why it may possess genes from other bacteria, and that skews the results a little bit. But for chromosome 2, you can see there's a clear jump between a ratio of less than 1 to more than 1 at the origin. And theoretically, the other place where it switches from more than 1 to less than 1 could be the terminus as well. So our final method was for hypothesis 2, which said that essential genes tend to be closer to the origin of replication. So as I said, essential genes are genes involved in replication, transcription, and translation. And we divided the chromosome into 3 sections, and we use it relative to the period of replication of origin. So it's kind of small, but as you can see, we have the 3 period of replication origins for chromosome 1 and the 5 for chromosome 2. So for chromosome 1, we wanted the period of replication of origin which had the most percentage of essential genes and most percentage of rRNA genes. So as you can see, in chromosome 1-3, we have the most rRNA genes and the most essential genes. And for chromosome 2-4, we have the most rRNA genes and the most essential genes out of all of those period of replication of origins. So that further substantiates our theory that the third period of replication of origin for chromosome 1 is the functional and true replication of origin, and the same goes for the 4th period of replication of origin for chromosome 2. So that was my poster that I presented at the American Society of Microbiology, and yeah, so it's based. So the poster is kind of like a condensed research paper in a way. Like you could write many pages on this research project. But the point of a poster is to condense all that and visually show all your methods, your findings in a very easy way to understand. So as you can see, I took these diagrams and put it into the presentation for you all to see. And so that was me presenting on the battleground to present. So this conference, which was called the American Society for Microbiology, and it's the Texas chapter of that was held from November 10th to 12th at UT Dallas. So just recently I went there. And at this conference, it's not just presenting your research. So you can hear other people's presentations as well. So there are 2 types of presentations at this conference. There are oral and poster presentations. So poster presentations is kind of like a grown-up science fair. That's what I think. So you have your poster there, and you stand by poster, and then their judges who are just faculty members at the many universities we have in Texas. And they come and ask you questions and judge. Fortunately, I didn't qualify since I wasn't an undergraduate or graduate, but it's still a very good experience to have. But in addition, you can listen to formal oral presentations, which are 15 minutes long approximately. And you can hear everyone from undergraduates, graduates, PhDs, even doctorates. So it's a very good experience. Definitely some things we may not understand some things, but I think that's good. You get exposed to the whole world of research just firsthand, you know. So that was a really good experience for me. It really opened my eyes to, you know, what's current in research, and it's actually very interesting, even though it's very complicated. But I mean, I like how it's so complicated, and you have to constantly figure out like what they're saying, and then you also have your mentor, so you can of course ask them questions. And yeah, like I said, presentation for professors. So for future work, so I really believe that, I mean, research isn't just you do it in the summer and you're done with it. It's not. Research, I think, is a little different than an internship. An internship is like you do an internship and you're done with it. But research, I think, is more of a long-term thing that you're continuously expanding on, continuously discovering new things. So I plan to expand our methods, our 3 methods, to other species as well, and verify that these methods actually work in correctly identifying the origins of replication. In our case, we did correctly identify the replications of origins because another master's student conducted a wet lab experiment, which is like they biologically confirmed that these sites are the origins of replication. So we were successful using just bioinformatics methods, which I think is just incredible that we can use computer programs and number analysis to find replication of origin. So that's pretty cool. And we also plan to design a pipeline program. So a pipeline program is for like what we're doing is where we will input a complete marked and sequenced genome of an organism, and it will just output the replication of origin without individually doing all the methods. It's just embedded in the program. So that would be a good challenge to have. And also, I do plan on, after having done all these things, that will be substantial enough for me to write a research paper. So I do plan that in 2017 or next year, after winter break, that I start to write a research paper on what I've done. So thank you very much for listening to me. And anybody have any questions about anything at all? You can ask me anything, like how many times a week or whatever you want to know. Yes.
What did you find particularly interesting at the conference that you learned?
So I think one was, it was a PhD presentation. It's specific, but it was really cool. It was about the microbiome of the human, which is kind of cool because we were talking about that in AP Biology. And so they had this cool thing. It was like, if I remember correctly, like when they tested patients who had like different sores or like eczema or stuff like that, and they linked it to a lack of bacterial diversity to the human biome. So I thought it's really cool. Like you're a walking, pretty much like you're a walking biome, pretty much. So that's very interesting. Other things, I mean, there's this very good talk by a professor. I do not remember his name, but I can tell you later. He had over 40 years of experience in just like one focus. It was the viruses, I think, and so that was just amazing. That he spent so many years and he had like over 100 publications, which just blew my mind. But it's just so interesting that even though it's a small like subset of biology, there's just so much research that you can do in it. So I found the conference really interesting and eye-opening. Any more questions? No, yeah, of course. Yes.
You had more than one chromosome. Do you think there's any evolutionary relationship between prokaryotes that have fairly complicated genomes like that, and eukaryotes.
Yeah, I really do, because I think, yeah, because it was originally a plasmid, that's what we believe, the second chromosome. So I can see that in the evolutionary journey, per se, that it could eventually evolve into a rudimentary eukaryotic organism. So that was really cool, actually. Only a small percentage of bacteria have a multipartite genome. So, and there are also various replication models that show how these two replicate. Because when you have a bacteria, you only have to worry about one circular chromosome replicating. But in this case, you have two, and you have to coordinate them correctly so that one doesn't replicate while the other one doesn't. So there are two models. One is when they just start at the same time, and then the one that's larger ends after the one that's smaller. And then there's another one, which I think is pretty cool, is that, so the first one's 3 megabase pairs and the second's 0.9 megabase pairs. So the first one starts, the 3 megabase pair one starts first since it's larger, and then when it replicates 2.1 of that, so you only have 0.9 left, that's when the second one starts. So they end at the same time. So I think that's very cool in investigating cell signaling with that and how they coordinate that.
So there's cell signals going on.
Yes, between the two chromosomes. Yes.
I just have a question. Do you feel like doing this has helped you know what you want to do after college? Like, do you want to explore something in this field or?
That's a good question. Yeah, definitely I would say, because I plan on majoring in biomedical engineering. So bioinformatics is actually a subset of biomedical engineering. So I'm definitely considering to focus on bioinformatics in college. But yeah, I think if you have the opportunity, you should really take it and do research. You'll learn a lot, and it'll really help you with your career decisions and even your undergraduate decisions. You know, if you do research and you do not like it, then you know that you should try to find something else or participate in other activities in college. But I really like research, so I know that I will definitely continue doing research as an undergraduate.
All right, thank you so much.